Meryl joined us earlier this May as she completed her MBA at Yale. She also has an MFA from the School of Visual Arts in New York. She is the co-founder of Chirps, which is a cricket protein company that you may have seen on Shark Tank. So Meryl is going to delve into some of the opportunities that we've identified in new protein discovery and the cellular ag space.
Thanks, Abby. Today I'm going to talk about how macromolecular imaging, computational protein design, and microscopy will help us develop new food products and ingredients. So as Leah mentioned earlier, as our population continues to grow and our protein demands increase, we need more sustainable sources of food. And as we probably already know, proteins are the building blocks of life and of food. And while we often think of protein as a dietary supplement that helps build muscle, proteins actually perform various functions within our bodies. Proteins are macromolecules consisting of one or more long chains of amino acids. Enzymes and hormones are two of the many essential biological compounds made of protein. For example, antibodies are proteins, and proteins working as enzymes break down lactose from milk in your lower intestine. And these are just some of the many proteins that exist with a variety of functions and potential use cases. I'm now going to dive into a set of visual technologies that are vital for discovering and creating new proteins. So first is macromolecular imaging, which allows us to view and discover new proteins. Macromolecular imaging is the viewing of structures of cells, often through a scanning electron microscope, which was used to create this image of an almond at 110x magnification. Macromolecular imaging allows us to closely examine the structure of various plants and their proteins. And when the ideal proteins aren't discoverable with macromolecular imaging, computational protein design allows us to create our own. Computational protein design is the designing and testing of new sequences of protein. By visualizing proteins on the molecular level, we can redesign proteins to fit new needs, such as specific flavors and tastes. And in fact, computational protein design is already being deployed by startups like AMI Proteins to create new protein-based sugar substitutes that are targeted at disrupting the $142 billion sugar manufacturing market. Macromolecular imaging and computational protein design are also being used to study and reproduce other enzymes, such as rennet, a key component in the thickening of milk into cheese curds. Rennet was the first ever genetically engineered enzyme approved by the FDA in 1990. Rennet is an enzyme found typically in the stomach lining of a calf, but thanks to macromolecular imaging and computational protein design, both plant-based rennet and lab-engineered rennet have been discovered and widely adopted by the cheese-making industry because it's purer, more reliable, and cost-effective. And since the average person, believe it or not, eats 39 pounds of cheese per year, it's important to have better ways of producing it. And as I mentioned, one of those ways is cellular agriculture. Cellular agriculture is the production of agricultural products from cultured cells. There are two types of products made from cell ag: acellular products cultured from organic molecules such as proteins like rennet, and cellular products grown from living cells such as lab-grown meat. And now microscopy, or the viewing and imaging of cells on the microscopic level, is crucial to the discovery phase of cellular agriculture. In lab-grown meat, for example, which is another cellular ag product, first you take a tissue sample from a live animal, then you examine the cells and create cell lines, which you grow in bioreactors until lab-grown meat is created. For lab-grown meat, microscopy is being used to help companies find opportunities to reduce the need for growth factors. Growth factors are the proteins that bind to receptors on the cultured cells and allow the cells to proliferate, and are currently one of the highest costs in lab-grown meat production. But lab-grown meat has a lot of other challenges aside from high costs. For example, structure. Some companies in the sector have attempted to use bioprinting to build structured meat like steak, but there's yet to be a breakthrough in the industry, and we're currently limited as a result just to ground meat like hamburger. And so we don't expect bioprinting to be a huge part of the lab-grown meat market in the next 5 years. Furthermore, lab-grown meat faces an uphill battle with consumer acceptance. According to a study done at Michigan State University, 48% of people were unlikely to purchase lab-grown meat. But unlike lab-grown meat, we're already eating, drinking, and consuming new proteins discovered and created by macromolecular imaging and computational protein design in the cellular ag space. In fact, one company, Just, already has an egg replacement on the market, has raised $220 million, and there's tremendous opportunity for many other startups to enter the space. Macromolecular imaging, computational protein design, and microscopy are already being deployed, whether in lab-grown meat or to discover and create proteins. Our conviction is that lab-grown meat has an uphill battle and won't come to fruition in the next 5 years, but we will see growth in other categories, such as dairy and eggs. Thank you.